Meiosis occurs in eukaryotic organisms that reproduce sexually. It could occur in both plant and animals, but I chose to do it in animals. This stage is a two-part cell division process that produces sex cells with half the number of chromosomes as the parent cell. There are two phases of meiosis, meiosis 1 and meiosis 2. Before a dividing cell enters meiosis, it grows through interphase. This is when each single-stranded chromosome is replicated. In meiosis 1, 4 phases occur. The first is prophase 1. The DNA closed tightly, individual chromosomes become visible. Synapsis plays a part with homologous chromosomes. These tight chromosomes may have exchanged or replicated their genetic information through crossing over in order to create genetic diversity. As this phase continues, the spindle apparatus forms around the centrioles on opposite end. In metaphase 1, the nuclear membrane disappears and the microtubules form a spindle. The spindle fibers attach to each centromere and the two diploids attach to microtubules orientating from opposite poles. This is when the equatorial plate forms. Each pair will then line up on the metaphase plate. Either maternal-paternal homologue may orientate toward a given pole. This can also be called disjunction through the separation of homologous chromosome pairs. Non-disjunction could occur if the homologous chromosome fails to separate, if the centromere doesn't split. After metaphase 1, anaphase 1 occurs. The spindle fibers shorten and pull the homologous chromosomes in order to separate and move to opposite poles of the cell. Since the tetrads are still held together, the centromeres don't split. Each pole ends up with the complete haploid set of chromosomes consisting of one member of the homologous pair. At the end of meiosis 1, telophase 1 occurs. These pairs begin to uncoil and the spindle fibers disappear. Cytokinesis occurs, which means a nuclear membrane reforms around the daughter nuclei. Each daughter nucleus contains two sister chromatids for each chromosome, attached to a common centromere. Due to crossing over, the two sister chromatids are not identical. After meiosis 1 is over, meiosis 2 occurs. In this, a new equatorial plate is visible. The first phase is prophase 2. This is when the nuclear envelope breaks down and a new spindle forms. In metaphase 2, spindle fibers bind to both sides of the centromere. In anaphase 2, the spindle fibers contract and the sister chromatids move toward opposite poles. At the end of meiosis 2, telophase 2 occurs. That is when a nuclear envelope pinches in and reforms around the sets of daughter chromosomes.